Research undertaken over the past 4 years at the University of Canterbury is using a milk protein to create a three-dimensional biodegradable template that muscle and bone cells can grow on. The process is called bioimprinting and has the potential to produce some remarkable outcomes.
So bioimprint as itself is a very unique technique of imprinting a cellular shape into a surface in full dimensions of every detail that you could possibly have. My colleague, Professor Manal Kaisi, had a PhD student, James Moyes, who back in the days was looking at isolating individual cancer cells and imaging them and looking at, can we tell from the shape of the cell whether it's a cancer or not? It was reasonably difficult to image the cells at very high resolution using the atomic force techniques that they were using. They would basically destroy a cell. So he had this brilliant idea to make imprints of these cells into polymers. And once they're in a rigid form in a polymer, you can do a very high precision image. You can scan as long as you want to get those surface features that the cells have. A second PhD student came along, Fahmi Samsuri, who actually did this with several cells. And it turned out it works well if you have multiple cells. You have a surface that looks like you have cells grown naturally, but they're all just shapes in plastic. The third PhD student, Lynn Murray from the US, she came and did her PhD again with Manal Kaisi, and at that stage I joined the group as a postdoc. And she looked in depth at what happens when you put other cells onto these surfaces. From there it developed really, and what can we do with this? And given that the cells react, this is where Azadeh came in. What started it was us reading another scientific paper from a group in Taiwan that was building artificial conduits for nerves to grow through, and they were building these out of casein. They were making basically micro tubes, and they got rat nerve cells to grow through these and reconnect a severed spinal cord, which is amazing. They had one problem, and that problem was that the material, when they implanted into the rats cells react to the material. They try to get rid of it. It's an invasive piece of material in the rat, and the body tries to reject it. So we thought, if we put some patterns onto these tubes and make them somewhat natural and get the cells to be thinking that this is something that's supposed to be in the body, maybe we can reduce that rejection. So the idea was, can we combine the techniques that we have here that Professor Al-Kaisi has developed in terms of putting cellular patterns onto surfaces with biodegradable materials like casein. So the idea is to get tissue to grow artificially, for example, and for that to happen you need to give the cells environments that they're used to in the body, and Azadeh is trying to replicate these functions on plastic surfaces, or in this case biodegradable surfaces.
So this is the biology lab, PC2 lab. This is where we started the process. I grow cells on the microscope slides and then fix them and take the microscope slides to nanofabrication lab to make the master mold in PDMS. I dispense liquid PDMS, which is a polymer, on top of the cells. Once PDMS is dry, we can peel it off. It's a flexible polymer, and all the features have been transferred onto the surface of PDMS, but they appear as indentations because they are reversed. Then I mix casein powder, I dissolve it into a solution, and then liquid cast it on the surface of PDMS. And then once casein is dry, then we can peel it off, and all the features are transferred onto the surface of casein. One part of my work was to look at the biodegradability of the material. The actual material without cross-linking would dissolve in media or in water within a couple of hours, which is very short for the cells to attach to the films. So I looked at adding different cross-linkers to increase the degradation time of the material. This is the end result. And if we put them under microscope, the surface has raised cell-like features, which we call bioimprints. And this is an example of a single cell, which is 2,000 times bigger than the actual cells on these films. So from here, we take these casein films with bioimprints back to PC2 lab, and we seed and grow live cells, live muscle cells or other types of cells. These are not live cells, but they can give physical signals to the growing cells, and we believe that's how they can influence the way cells behave and grow. I am hoping that we can use this biodegradable material as an implant with surface features. So the surface features would help the cells to behave in certain ways, or even tissue such as muscles to regrow in body. And then the implant itself would degrade, so there won't be any need for a secondary surgery to take the implant out.
We have currently only used muscle cells, where we know that they can rearrange into fibers and they can form tissue. But there are lots of cell types. What is probably the holy grail is to look at stem cells and see what stem cells do when they see surfaces. A lot of the plastic material that we used to use 50, 70 years ago was actually made from casein. So it's historically been used to make buttons and other objects. So it is a renewable form of natural bioplastic that people knew about, but because oil-based plastics became cheaper and it was easier to manufacture these, they have sort of, casein became obsolete in a way. We can say our surfaces react exactly the same way as the plastic that you've been using so far. So for us, it's a good thing that it doesn't have any different properties. From an engineering point of view, it's interesting, and it's more or less a renewable material. As long as you can get milk powder, you have lots of casein, and then you can make lots of plastic. So there's a whole array of research that has spawned out from this, and it's really a fascinating area. No one else in the world is doing that.
This program was made with funding from New Zealand On Air.